Well, good afternoon, everybody. I'm Lorenzo Veschini. I'm working with James Glazer at Indiana University, and we collaborate on several projects regarding model standardization and other stuff and integration. Let me see if I can put up. Well, no, I don't want that. I'm fine without that. Okay, so today I will try to guide you through a guided model building exercise of, from the coding point of view, very simple model, but then very interesting to explore. And we will look at a little bit on how we can explore it if we have the time. So without further ado, first of all, I wanted to introduce you to a little bit of what is that we mean when we discuss vascular angiogenesis. And so I thought I would put a few slides about microvascular biology, a little bit in a nutshell, let's say. So when we discuss vascular microvascular biology, we discuss the blood vessels and also the lymphatic, typically in the human body. And these are kind of widespread, as we will see in the next slide. And they are broadly divided in two big systems, the macrocirculation and the microcirculation. The interesting point is that in the macrocirculation, we have certain function. The macrocirculation is the one that typically you view in this kind of diagrams. Only the big vessels, like 3 centimeters, 0.5 millimeters, like the aorta and the big conduits from the heart. And the main function is transport of blood, like flow, and to and from the microcirculation. But at this level, there is no gas solute exchange. In the microcirculation is where some more interesting bits happens. And this is interesting to say in terms of virtual tissue modeling, because the microcirculation is probably the one we will deal with. Many models deal with this aspect, but when we think about tissue, we are at this level. And at this level, we have blood vessel doing a regulation of gas and solute, a regulation of tissue development, angiogenesis, that is the topic of today, of this exercise, immune cell trafficking. And these are just fun facts about the blood circulation. And this is about flow, so it's not probably very relevant today. A little bit more about blood vessels. These are, it's a very interesting paper that you can grab here. If you grab the QR code or you just search for Bell et al. 2017, they have stained human embryos for vascular and other markers, and they were able to build a 3D, three-dimensional atlas of these structures. And this is, if you see them, I always like to show this image because they are very, very good. They show how much blood vessels without this tiny white structure against the dark background, so there is nothing else but blood vessels represented in these images, are really, really diffused throughout the body. Some more quick fact about microvascular and endothelial cells. So the endothelial cells, which actually are the inner lining of all blood vessels and the main cell type in capillaries, are 20-25% of all nucleated cells in human body. And the cardiovascular system, including the heart and the blood vessels, the big, the larger, and the more circulation, is the first functional organ system in the human body during development. Endothelial cells are main cellular players during vascular angiogenesis, which is the formation of new blood vessel, during tissue development by angiocrine signaling, and they are gatekeeper inflammation and immunity. And when we discuss disease, localized endothelial cell dysfunction is a hallmark of several pathology, if not many, including cardiovascular disease and cancer, if not all. Okay, this next slide shows even further the concept. If we look at zoom in on a particular organ, in this case is the developing lung that has been stained for structure like the distal epithelial bud, so the forming structure of the epithelium that will absorb the oxygen, exchange gas, so the oxygen, and the proximal epithelial bulb, which form the bronchi, and then the blood vessel, which comparatively you can see they are really in close juxtaposition with all the other structure. And this is, again, you can find this information in this paper. A little bit more about vasculogenesis and angiogenesis, which are the process by which blood vessels are formed. Vasculogenesis is the formation of new vascular network from newly differentiated endothelial cell. It's particularly relevant in during development, but it's also happening in adult life. Angiogenesis, which is the prevalent form that happens in adult life, is the formation of new vessels from pre-existing ones. So capillary network constantly reformed, like together with tissue, which regenerate and repair, or even just in homeostasis, they renew. And there are different types of angiogenesis. The most common and most often studied is sprouting angiogenesis, and non-sprouting angiogenesis as well, like intersubjective angiogenesis and other kind of angiogenesis. So if you look at this diagram, we see that vasculogenesis is initiated by creation of endothelial cell via mesodermal precursors and angioblasts. So this is happening during embryogenesis. And the primary capillary plexus is formed. Then in human and invertebrates, the two main cardinal vessels are formed, and then they fuse, giving rise to the big vessels in the human body, like the aorta and the big vein. And then all of those, of course, are, while the tissue are developed, form a tiny capillary network that look like this into each and every tissue of the body. And through this process as an early phase, let's say, where vessels are formed, but then they are pruned and remodeled and surrounded by other cells, and this is called remodeling. And this yields a mature vascular system, which is typical of homeostatic tissues, while this is typical of juvenile vascular system or even reparative tissues. Okay. Okay. These next slide are nice YouTube videos which show you the process from two different angles in the experimental model. The left one is a zebrafish time-lapse imaging, where they have taken engineered fish, which are stained with particular fluorescent proteins, which are actually express their fluorescent protein in the genome, so they can be visualized. And if I rewind the video, we can watch again. It's not really immediate to appreciate in individual cells, but you can see that cells form protrusion, they extend, and this process is guided, and but is also self-organizing. And it's very ordered. If you can see, many vessels are formed from the primitive one. This is an example of angiogenesis. And you see the parallel vessels are formed, and eventually they anastomose to form a primitive network out of a bigger vessel that was pre-existing. In the second movie, that is something we will look at again, that is more interesting for our lecture today, for our exercise. We are looking at endothelial cell. You remember we discussed them, and how they behave in vitro, in a petri dish, let's say. And so there are experimental systems where you can use special matrices called matrigel or derivative of those. And when you put endothelial cell at a certain density, they self-organize to form structure that they resemble primitive endothelial network. And in some instances, they can even be patent, so having a lumen inside. And if you can see from the video, this process is kind of patterned and repetitive. So you see that they forms a certain type of structures. And we will see a little bit more of that again. Okay, and this happens over 12 to 24 hours, depending on the experimental platform. You can observe different times, but this is more or less the time frame. And there are many other experimental platforms to appraise different type of vascular angiogenesis. But again, this is the one we mostly interested in today. Okay. Vasculogenesis has been modeled before, and actually what we are doing today is trying to look at something that has been published before by the Glaser group. But these are example papers that are a little bit more recent, and they look at different ways of modeling vasculogenesis and angiogenesis. And you can look at that and grab those QR code, or anyway, the slides will be available, and they are already available in the material, student material folder. The one that we will actually look at most today, and especially we will try to not to reproduce it in full, perhaps this will be an exercise for you if you want to do it, is this one, chemotaxis-based models, and this Merckx et al. 2008. And if you want to grab the QR code, it's handy to have it, not necessarily for the lecture, but for later on. And anyway, you will have everything available for your review. Okay. Now, the exercise is about trying to build a model of angiogenesis, and I promise it wouldn't be difficult or complicated. So I'm taking some time to talk about the preconditions. So what we observe in vitro, again, out of the movie we have seen before, we see that in the first 3-6 hours, endothelial cell migrate, so they move in this experimental matrix, above this experimental matrix. And they keep a round shape until they reach the range of interaction with other cells, whereby they elongate and they join together with the other cell. Eventually, the cell form a continuous multicellular network, and this is kind of evident from the movie. No advertisement is intended about the company or the movie. Is a free one on YouTube. The one important thing to notice is that the endothelial cell in this particular assay stay above the substrate. So 2D modeling is appropriate for our purpose. And this is what we will do. First thing, we may ask some questions about how we do want to model, how the experimental model might relate to our model, and so which are interesting quantities to think about. So, for example, how do cell density affect tissue configuration? So we say that we are in an experimental system where we put endothelial cell on top, but yeah, we may think about a confined area, but then how many of those cells we want to put is always the same, it changes. Experimentally, we know that it changes. So modeling wise is good to know. How does cell adhesion affect tissue configuration? So we might think about two types of cell adhesion, maybe cell-cell adhesion or cell to matrix adhesion. And in our model, we might think that we have to handle both in some way. So these are important aspects to take in consideration. So how does field decay? Here in the slide is a little bit opaque. So we assume that there will be some molecule that might guide the sprouting. We know that in real angiogenesis, the main driver of angiogenesis, or one of the main drivers, is VEGF, vascular endothelial growth factor. And in our model, we want to model that. And so we want to ask how much is produced, how much is decay in the medium or the environment, and how these parameters affect our growing network configuration. Once we have implemented a model with another of these parameters, how can we predict, in fact, angiogenesis, the one that happens in vivo? or in vitro from a certain parameter setup. And if we do, how can we guess what the parameters are and what they mean in real life? Okay. So one interesting aspect, and that's why I wanted to put this slide here. I inherited and I liked it a lot, is about how the interaction between an evolving cellular assembly and a gradient that induce chemotaxis. And we have seen this morning, Pedro explained quite extensively how chemotaxis work in CompuCell and in general, and how this interaction might yield asymmetries that might yield morphogenesis. And in this case, we can see that in this slide, it's highlighted how initial random fluctuation in the boundary of a tissue arranged like that might change the steepness of the gradient of the molecule that is produced by these cells. And if these cells are then sensitive by chemotaxing to this gradient, then this might amplify the process and create structure that then become more organized. Okay? And this is the basics that the model will exploit a lot. Okay, so now the next step is actually the exercise. And I might not have told you a lot now what we want to do. First of all, we want to know what we want to put in our model, and then we can try and build it, and then we can see how to do it. Anyway, in the student material of which you have the link, and I will provide it as soon as, again, as soon as I stop talking, you will find a fully developed simulation that we will use today. So if you don't feel like following along, coding along, you can just use that. Okay, back to the basics. About objects, what do you want to represent in our simulation? We want to represent endothelial cells. We say that they are the mastermind of all the process, and they are guided by VEGF, vascular endothelial growth factor. Then we, like in all CompuCell simulation, like in all simulation, we have medium, so the work, the environment, the substrate. Behaviors that we want to encode, and the endothelial cell will want to move, and they will want to move probably randomly. They will have a volume. They might or might not elongate. They will adhere to themselves and to the medium, and they will respond to the VEGF field. And the VEGF field itself will diffuse, will be produced, will be diffused, and will decay. In our case, we will have the endothelial cell produce the VEGF field. And that's why it's relevant, the slide that I showed you before. About interactions, we have, again, endothelial cells will interact with the VEGF field by secreting VEGF and by chemotaxin. And the endothelial cell will interact with other endothelial cell via adhesion. And here we have an important aspect that we want to have chemotaxis blocked, chemotaxis to occur only on non-adherent surface, not where the cell interact with one another. And this is something we can implement in CompuCell. And then the dynamics is our usual that we've seen along the course. So we use the pot system and the diffusion equation for the fields. Okay, now that's the exercise. Build the angiogenesis simulation. So I will stop talking for a second. First I will read the slide, and so we can kind of try to do it along. And then I will put again the link to the material, so if you wish, you can download it directly. And I don't know, I will give you 4 or 5 minutes to think about it, and you can send me thumbs up, thumbs down if something is not working, etc. So point one, we want to build a 2D 100 by 100. The one that you find in the slide in the material is bigger, is 260 by 260. Your choice. It should work anyway. With periodic boundary condition, and you can use a blob initializer. The one that you will find in the material has a few ways of initializing. So, but the default one is a uniform initializer, but you can find the blob as well. Then you want to define the cell type, endothelial cell, and define a VEGF field and choose DiffusionSolverFE. And I think you can do all of this via the wizard. And you want to select these properties: behavior, contact, volume flex, chemotaxis chain, field boundaries to periodic. And you want the cell to chemotax. So you want the cell, the endothelial cell to chemotax to VEGF. And then you need to set a few parameters regarding these behaviors. So in the next slide, I will give you the standard parameter that works. And those are also in the already preset in the default in the material student material version. So if you want to take a few minutes to try, and or to ask any question if you have any while we pause for a second for the exercise. And in the meantime, I will put again the link to the material. Yes, to answer at the yes, the slides are in the Google Drive. They are possibly not the most up-to-date, but the key material is there. This simulation just needs to set up the XML side of things. It doesn't need to set anything else, a Python code, but you will see that in the student material there is a part with the Python code, and that we will use it later for analysis or for reporting, for plotting. So you see already here in my version there are some plots, and this is what we are going to see in the simulation. To set the chemotaxis, you need to set the LambdaChemotaxis and the parameter for chemotaxis. Yes, it's missing there. Really, the next slide will be most a suggestion for which parameters are optimal to get network happening. So, okay, those that are done, why don't they start putting a little bit of example just to see what they are getting. So I'm curious to see if all get networks like the one we see or other things. Like Robert sent us these examples, which looks great. Okay, how many of you that I see that a lot are 70% of you are done. How many of those that have done are getting a network similar to the one that Robert has put in? Oh, sorry, Eudes then has put in, and who's getting something else? Just put in chat. Okay, after the collision, the program crashing shuts all down. Okay. There are a few reasons for, yeah, exactly. Yes, I suggest the XML there might be something with XML. So this is probably something relatively trivial. So I will move forward. The only thing I want to show in the next slide is, and this is an ongoing exercise, which are the parameters that typically yields a network similar to the one that you see here. And the one that if you put them as a standard, they will give you a kind of similar to that. But interestingly enough, the one thing that we would like to ask is, okay, this is not this is cool when we look at the example we saw in the in vitro assay, and it seems like, oh, fantastic, it does that, and actually I can replay that for in the interest of all, if you want to see how it evolves. And I start from a square lattice like this with a few cells. I give a little bit of boundaries. It's interesting because we can discuss this a little bit later if we have the time, how we handle boundaries. In this particular example, you will see that the periodic boundary for the cells will end up having the bits being interconnected. Okay. Oh, is there no mitosis involved in the process of angiogenesis, or are we choosing to ignore it to simplify things? In this case, if we look at the experimental system and the timeline, there is very little proliferation. But in angiogenesis, there is a lot of proliferation going on. So our system is especially good, the model is good at modeling morphogenesis in terms of assembly of endothelial cells and look at if this is guided by the VEGF. So it's a sort of initiation of angiogenesis rather than the process. When the vessel needs to elongate, they will need to proliferate, and there is a whole process that we don't model here. Let's explore it a little bit, because if it was all like this, okay, we're done. It's also so good. We are accidentally found the perfect parameters that repeat an in vitro experiment. And we might ask is actually true, but we will ask ask this question in the next slide. But first, perhaps we want to try, and this is an exercise for you. And this time, I want see some images in the chat. All of you, those that they have a running simulation in front of them, please play around with, choose one of those, the one that you like best by the name or because you like it, and try to change it according some meaningful way in a few tests, and show me perhaps some interesting results. Do you see always the same thing happening? It's always the same, whatever you do, something is changing. And yeah, I left it to you another 2 minutes because this is quite fast to realize, to make it happen if you have the simulation running. Yeah, I repeat what James is suggesting in the chat. You can change the diffusion length of VEGF, which is very interesting in this model. And of course, you change it by changing the diffusion coefficient of VEGF. And you can make it longer, very short. What happens to the networks? Does it form at all? So I'm not seeing things in chat. I will play around myself. I will pick my favorite one. I will make it small. Not so small. Let's set the coefficient diffusion to 0. It is as suggested. Diffusion.
So I was going to break in and repeat what I said in the chat.
You
If
do
you
well.
change if you change the diffusion length of VEGF by changing the diffusion constant, for example, or the decay constant, the pattern will change and changes in interesting ways. One thing to look for, does the pattern change quantitatively only, or does it also change qualitatively? The initial density of cells in the simulation makes a difference. There are interesting pattern changes for that. And the surface tension, if you change the cell-cell adhesion, that changes the pattern too, and you can get phase transitions in the pattern from that. Also, if you turn off the chemotax-towards medium option, you could get some different results as well. Those are all worth playing with to see what you get.
Yes, that is the basic idea, the autocrine production of VEGF by endothelial cell, which occurs, and of course is not necessarily the only force, only driver of the process in vivo, but in the specific assay, there is a mixture of exogenous and cell-produced VEGF that generate the pattern. So in these cases, the modeling choice was to only consider the one that is secreted autocrine by the cell itself. Just changing some parameter at random just to show you while you do it yourself, what are some options that might emerge. Let's see a few options. I will try one more, see if I can. Do one of the things that are suggested. So I'm removing the Chemotaxis-Towards Medium. some ways for this. Yeah, different button buttons. These fields. Okay. That's very highly constant. It's not extremely high, but let's see what happens. Good. Well, I think we have explored a little bit of the funny bits. See, there is some other comment in chat. Lovely, the one that good. Yeah, nice example, I see. Really, really nice. Rita, you might have perhaps a little bit too few cells, which is exactly what Prof. Glazer is suggesting right now. You need a little bit more of cells, so your initial condition needs to put more cell in. Okay, let's go back to the slides. A second, get the replay a bit. And we can move forward for the past 20 so minutes. So we have a few minutes for chit-chatting. So, and meantime, you can, of course, keep playing with the simulation. We will do it in a little bit more systematic way in a moment from now. So, so far we have watched the behavior of simulation, and we have explored for a second the angiogenesis simulation, and already we see that by tuning a few knobs, we can change the behavior of the system in a dramatic way. So how do we capture these in a more systematic or automated way? So dependently on our simulation, is important extracting results from our simulation, numeric value that are easy to plot and so on. So typically we can have population dynamics matrix, so cell count by type, the total, the variance, the variance over time, the fraction, derived matrix, that transition rate if they differentiate, all sort of things. The cell shape of these cells, if it's important, what we are trying to investigate, the morphology of multicellular assembly, like what we are doing now, looking at patterns, how they connect and how to relate, how connected components interact, how they organize in space, how do they move over time, so displacement, we can track center of mass or other bits. how do they interact with the chemical environment, with our gradients and fields, how much they uptake, how much they release, and how much they release in response to eventual stimuli. How they grow, how do they change, mechanical properties. In CompuCell, we have some limits, but of course we can do something, and there are proxies that are kind of interesting for pressure and for junctions. We have links, so we have a lot of interesting bits that can be captured. And then, of course, if we are interested in validation in terms of training models, etc., we want this kind of typically more standard boilerplate metrics. At the same time, when we want to actually tell our story, so we have seen that, for example, some of our parameters make the structure change dramatically, and we want to report it somehow. And so plotting becomes important. Perhaps this is not really a key slides here, but since we will discuss this aspect in the next week about the importance of really reporting your data and putting it in a way that are useful and more at a higher level of interpretation, plotting becomes really handy. You can really reveal dynamics, it can help detect problems if our simulation doesn't behave. It supports comparison between different simulation, quantify outcomes, improve communication, and of course, it guides model refinement. So in our case, we have our simulation produces, as we have seen, this kind of patterns. We can easily export this kind of image via the built-in system in CompuCell3D, or grabbing snapshot, or writing them in Python. So this is an example of an image that might well be a direct export of CompuCell. And what we want to know about our blood vessels, so which are interesting metrics. Okay, yeah. So yeah, that we are here, we might take a look at that. And let me see. I have to interrupt 1 second my presentation. And yet that we are here, when we want, let's take this down for a second. When we want to create a snapshot of our movies, first of all, we have to remember one thing. We have to let them know. It depends. Sometimes it happens, sometimes it doesn't. It depends very much if you have done it already. You have to set the scene for your snapshot, and you do this by hitting this camera button. This essentially frees the kind of the idea that is here in your plot, the sizes and this kind of thing. Once you do that, you can go in your setting, which I believe are those. No, they are not those. The range I'm looking at, but I most often look at setting around here. References. Style config, no, not this. I need to look at the other ones. Sorry, I'm not very familiar with the latest in settings. I'm not sure, and I'm in the right. This is the newest version of CompuCell 3.9, 4.1 of the newest 4.9. So I'm not sure I'm getting there. Oh, here is probably, yes, this is the place I was meeting. So output. So if you want to export movies, you have to make sure that this is well set. Otherwise, you will get an error in your this will be relatively silent because it will happen here into the terminal. It's not silent, it will happen, and you will see clearly if you look at your terminal, but if this is not set properly, you will have to do it, otherwise you won't have the chance to actually take advantage of this, which is once it tells you, well, update the screen every 10, you see that my simulation looks a little bit not very fluid, but it's not because it's slow, it's just because I'm showing only every 10 steps just to speed up a little bit. And then we can ask the CompuCell to save image every dot MCS, specify a resolution. You might tick this to have a movie being saved automatically every a simulation stops, or otherwise, if you want, you can take your sequential snapshot here and actually have them make them a movie yourself. You can specify specification here. You can create a movie of your current simulation, and you can show it in your file browser. And this is it. I'm not sure, since I'm not on my primary laptop, if I have example to show you. I hope I do. Let me see. Yes, okay. Can I show it those that the one that are of this simulation, because I didn't activate, but this is the kind of things that happen. You have you show where you want those things to be saved, and this happens if I'm not wrong in the setup where you put everything. So workspace where you want things to be saved is this one. Okay, and here you will have each times you save and run a simulation, each time you run a simulation, a new folder will be created with your simulation itself, and actually you can use it as a snapshot. And then if you have set up your properties as you wanted to properly, you will have this kind of folder that shows the field that you have available and the PNG, the PNG of your simulation, your snapshot. Here you go, like this. Not sure this will be interesting, but yeah, okay. So this is how it looks like when you save a snapshot. So you can really have export movies and PNG files easily, and that's it's very convenient when you want to do some post-hoc analysis. Okay. So let's go back to the presentation. And the reason why we might want to have these images. Once we have our images, we can analyze them as such. And one interesting aspect in vascular biology is looking at the shape of the network. So we can make a skeleton, analyze how many branching points. So this is typically an index of complexity of the network, how long it is, so how organized it is, how many branch per main strata, if main strata are evidence, and this kind of thing. We can have as a image-based matrix, we call it distance for medial axis. This tells us essentially how thick are our structure. And this might be informative in terms of vascular biology to tell about the hierarchy and the perfusion rate. Sorry, not the perfusion rate, the perfusion capacity of our network. So assuming that large vessel will bring more blood, not always the case, in fact. Then we might calculate a surrogate marker, which is not exactly the network as formed, but is the structure that are forming our images. So the lacunae, the spaces that are in between forming of formed structure, and that give us a sense of the network coverage. And this is interesting again to have an idea of perfusion. So for example, we know that by definition, almost, or because it's biologically efficient, not many cells lives farther away than hundreds or few hundred microns from blood vessels. That's because they need to get the oxygen and the nutrients. So in one way, in these surrogate networks, lacunae that are too big are diagnostic of a network that is not really covering our space in an efficient way. Okay, so these are thoughts that are very related to this particular experiment. I'm showing you the conclusion of a few discussions that we had with a group of people that are working toward these ideas. And so you have to think that when you build your model and when you analyze, when you think about your model, you might want to know as early as possible which metrics you will use to actually appraise how valid, how faithful to the biology, or how comparable to others is your model. And so metrics are important for this. Again, another important issue about metrics is what if we want to address an inverse problem. For example, we want to know, given a simulation that we believe is biologically meaningful, and it's run by a few parameters, and but we don't know which parameters could be good to actually represent the biology that we see in experiments. And so we might want to do the reverse problem of, given the results, given data, experimental data, how do we infer the parameters that might reproduce those data in a simulation? And so when we do that, we need a matrix or more. And then should we pick one to capture them all? If we do, which one is the best, or we should combine them into one number so optimization algorithms work better. These are interesting topics, and some of those will be covered in the next week, which is more advanced. But this tells you how important is right now, even when you're starting building a model, which matrix you will use to measure it. And this is often dictated by which data do you have available. And that's why we started with an experiment that we know that creates this kind of structure. And actually, if you have met some blobs into your exploration, these are also reproduced experimentally by excessive cell density, of too little VEGF, and things like that. Okay. Yes, absolutely. I then ask for a couple of students a little bit more support to have it work, for sure. And let me see. Oh, this is the last, I think, that is a little bit theoric. So it's important that we think. I will go fast on this today, and I will probably take it again next week. So when we choose matrix, and when we address the last question, which data are available, we should think that biological data are terrifyingly sparse, and they are also very variable. So this is the example of, if you have think about a little bit of the network we have generated today. And this is an experiment from one single sample that if we look at different spaces in this image, we already observed very different structures. So when we model, we have to think about, okay, there is biological variability, and there is simulation and simulation variability. and experimental variability. So we have to compound all of those when we really think about matrix. Otherwise, we might never solve the problem or overfit the problem to an ideal system that is perhaps not very realistic. And I guess there is one thing that I might do next week instead of doing it now. So I give 5 minutes to help Paul to be on board and having their simulation branching. I have prepared a little digression on a Python code that serves the actual analysis and yields results for this kind of a simulation, but we can instead talk about this next week. Oh, nice clusters. And so, no, well, that will take a little bit of time. Since data wrangling is the topic, I might just go. They are very simple script, and I've already included in the material. So those of you that are into Python, they will really find those kind of very intuitive. And so you can just use the student material and think about, there are examples inside that you can use. And so you can prepare to the next lecture about those with those examples. Also because today perhaps not many can follow up because it needs a little bit more of imports that are not included in the standard CompuCell installation. So, but I can use the time actually to have a little bit more fun with, so perhaps I can give some ideas on what we have found in doing exploration over this angiogenesis model. And so it's food for thought if some students want to really play around and actually go and try to reproduce the MERP paper. So if we look at, we have analyzed our images as I shown you before, and we have analyzed, so essentially the Lacunae. And when I thought about Lacunae, I thought about these. And when I talk about network width, I discussed these aspects of how distance each pixel is from the center and the average of those, and the network length is the sum of these pixels, and the number of branches are these. So we might encounter some of the metrics. It's just a selection of those that we analyze. So it's just to show you the interesting bits and patterns that we can really find. And then you can try to reproduce those yourself. So here, and also highlight how much plotting can be useful. So here we have taken as a matrix the linear saturation coefficient. That is one of those that perhaps you don't change very often, but we find that in terms of number of region, which is what we need with the kind of thing that we need, we want to simulate, this happens into the low ranges of these saturation coefficients, while increasing that tends to generate blobs. And if we just observe how the mean network with matrix changes, we see that it goes more or less together, it increases with the blob. So essentially our distance from the skeleton of the network increased because these blobs are definitely bigger than each individual, wider than each individual strut. So, and we will see later a little bit more of this. If we change another parameter, that is the VEGF degradation rate, and we look at what it does to the network width and to the number of region, we can see that it tends to generate very blobby-like structure in the very low range, while the network becomes denser in the higher range. And in terms of the width, again, the blob look much wider. But in general, in the higher range, we don't observe huge changes. Perhaps there is something here, but not much. LambdaChemotaxis, we see that there are again, changes that are reproducible, number of regions with low LambdaChemotaxis, there is nothing. Oh, consider that these parameters, I'm saying I'm varying these, I'm varying these, I'm varying these. The strategy of variation in this particular case is one at a time, which means that starting from those parameters that we have in the slides, we change the one that is indicated here. So the basic parameters that are written a few slides before, minus a change that is represented here, just for clarity. So only one parameter changes in this estimation. And here again, here we have the energy contact. Makes somewhat sense with number of regions. There are ranges which are different. And it changes a little bit the mean network width, not by far. We are talking about 165 versus 2 pixels, so it's not huge, the difference. Okay. Other interesting thing that can change. Here we analyze 2 parameters changing at the same time, and we look at the average thickness of the struts, and we see that it goes down with change, and we have here representative images that shows us how things move. Okay, no, it's just the number of lacunae. Okay, it's the usual stuff. And here we have a little bit more of replicates for each condition. And here we change the cell density. You remember it was suggested during the class that we might change that, and you see that at least in the condition shown into the exercise, we have different things happening, and starting from different densities. And this is actually really, I've done a few matrigel assays in my life in vitro in that manner, and this is actually something that you see. If you put too few cells, they will form non-connecting networks, and if you put the good amount, they form this level, and if you put too much, they form blobbish-like structure. Depends also from the VEGF conditions. A little bit more of the same, I would say, at this point. If we change 2 parameters at a time, what happens to our parameters, number of regions, and length of the network. If we have some target matrix in mind, doing this from these 2 plots, we might say that we want, I don't know, just for be funny, about 50 regions and the network to be relatively long, we might want to pick a region of this plot in this area here, perhaps. Okay. And nothing. That's, I think, pretty much it for the slide. So we can leave the last 10 minutes for debugging and playing around together. And this is the exercise left in slide for you if you wish so. There are some notes for doing that. Reproducing Merckx et al., the paper that I highlighted before. They show certain, with some nomenclature that the parameter, slightly different nomenclature of the parameters. And you see that there are a few that they analyze, and they often eventually find that the standard parameters, so more compound parameter might be more interesting to vary together, but please read the paper, it's very interesting, and after the lecture it might be that you really want to read it with a simulation at hand while you try to really reproduce each and every figure. And so this is for the questionnaire, but let's stick around for playing around with the simulation. So I leave this on just if you need to, and I'll then move to the chat and see if there are questions and if we can do something in the last 10-20 minutes. Okay. That is, I can show you that. Yeah, it's something I wanted to look at. And I told you that there is no Python really in this example, but in my example there is. And one of the things that is actually would be interesting to look at is one, where do these two fancy plot come from? And they come from the start function, some other iteration down the line. I think plotting has been addressed or will be addressed. Is for sure interesting. It has been addressed. I try to address it externally for the external plotting, but also for internal plotting, we can do very interesting stuff. And the example give you the example that a prompt for that. And then there is, yes, schemage morphology, schemage measures, SciPy and image that are not into the standard installation that comes with CompuCell. And so this will fail if you try to run the example. But the one thing that you can use to fix it and use it, just the XML part, if you would like to do that, I think you can just comment these two lines into the main script that is not really used here. And if I'm not mistaken, I shouldn't crash everything. I should have the XML part of the simulation. Let me confirm that with you. No, it's not. Probably I had something important in the Python as well. Okay. So this is not working, and it's actually close it. Let me go back again. This should run just to see that is not my computer that is frozen for some reason. No, it worked nice. It's just there must be some interesting Python code. Okay, so here the schemage part is used in this area where you use it to measure things. And therefore, if you just comment that area away, even if you will lose some features. Let me see. Yes, of course, you need to comment also the plotting away. So essentially all the step becomes meaningless. You could even put a pass instead of doing this. And then you remove the imports. In principle, now we should be working. Yes, now it works. Okay, so if that was blocking your example is that. So, but if you want to actually keep it and use it, it's actually very useful. The only thing is that you have to pip install this SciPy and schemage in your environment, and that will work perfectly. And actually something you can do is worth doing for the next time. In the source code, there are no component wall apart from a scene. Yes, that's fun, actually. I wanted to play around with that with you today. Yes, exactly. Or pip install scikit-image. If you are done the conda way, you are very safe. I'm sorry I didn't give you a YML file to install a full a full package for that. For the next lesson, I will for the next lecture I will do it because these packages are important. Okay, so let me start again my simulation and see if I can do, because I wanted to show you the whole thing. That is interesting. Okay. I think Zoom is making my computer at the test today. Okay. Well, it's already good. Let me see. Might it be that you have too little cells? Try the parameter that the default parameter and see what happens. So the one thing I wanted to show you now, I think is interesting, so I can make this bigger. I regret having stopped that. I don't understand why. Okay. What I wanted to show you is that under the current standard condition, the one that defined the model packaged with, you will see that the sprout extend. You can see one coming up from below, it comes from above, and then there will anastomose, etc. Actually, it's interesting that for this thing for analysis is important, so you will have to consider that for analysis. But this is another pair of shoes. But what if we don't like this? We really are interested in studying a more constrained environment when things cannot happen, that they join together, is not a sphere or something like that. So there is an example directly bundled into the one that I have uploaded, and we can do, if we comment line 151 to line 158. That's another, okay. I think Prof. Glaser will be able to explain that better, but if cell hits the wall, in some case they behave a little bit different. And typically, if you want to make sure to have a wall that behaves as you want, you create it explicitly. And there is an automated way that I didn't use in this case, but I wanted to have fun also as an exercise, because it shows you, you can choose, you see, you can compose bits in the uniform initializer. So what I've done here into the uploaded simulation, you uncomment everything that is building a wall and building a wall end, and you comment the region below, that is our previous one, and then we try and see that we don't crash everything. And I suppose the wall cell has been declared before in the simulation, and they are frozen cells. Okay. And in name contact inhibition, we impose a high energy between endothelial cell and wall. We don't want endothelial cells to stick to wall. They hate it, and they will stay kind of away. But to do that, we need also a wall type cell that we need to freeze if we want to avoid having to do things. And in this case, now we have a real Petri dish, right? the experiment is contained. The cell, we decide if the cells stick or don't stick to the wall. If we want them to stick to the wall for some reason, we might lower this energy perhaps to 3. Just lower. I don't know if that will make things work well, because I would probably do 4 just, but I'm not sure this will work anyway. But just for fun, no? And then I think I can get any question you have, guys. I'm happy to get anything. Otherwise, I can close it here. Let's say, well, now they can stick to the wall a little bit better in principle. And in fact, they do, unless they have a preferential contact with their pulse endothelium, with whom they have better contact. And you see, in this particular case, the stickiness don't prevail, but they remain attached. Then this might be your, in physiology, if you do a real matrigel assay, let me see, this may even be the case in the sense that really your cells don't stick to the wall. Typically, I'm not saying that they avoid it, but it's structured in a way that they won't stick to the wall. And so you might have this kind of structure at the boundaries where you have kind of long lines, let's say. And I think that was all for me today. So I don't want to overtime, first of all, but again, I'm happy to take questions, and I can stick around another 5 minutes if people have troubles with the simulation and cannot make it work. All right. So I think it's my duty to put back the last slide to remember you to compile the model questionnaire. And yes, I'm happy to ask any question.
All right, thank you, Lorenzo. So yeah, if you do have questions for Lorenzo, you can go and post them now. I know we did have those 2 students who would like to walk back and talk about how to get the initial cluster to branch again. If you have other questions, more general questions about anything we've talked about today, feel free to ask them now as well. But please do fill out the poll. I think we've already... Did we already launch it, James?
Yes, the poll is open.
Okay. Yeah, so please do fill out the poll.
Let me see. I didn't test it that, so I'll do it online. I don't promise that it will work because I have the blob.
So maybe Anna or Ritra, do you want to explain the problem you're having? We can give you permission to unmute.
Yeah, please do come back to me in mail and anyway. I'll be around. Grazie a te, Leonardo. Let me see. No, it didn't work. I'm sorry. At this point in time, I need to. I don't know why it didn't work. Yeah. Okay. So with the blob, I need a little bit more of testing. Oh yeah, that might be the problem. Radius a little bit smaller. Some 50 with maybe that should be better at least. That looks better. I don't know what they will do though. Yes. That works with the blob as well. So if you're starting with the blob, this might be a good configuration. You might make it a little bit bigger as well. Let's make it 80. And the gap of 3 may be even big, but let's see what happens. Yeah, with the blob, it goes this way. Okay. So if you want to just drop me a line in case it still doesn't work for you, we can look at that tomorrow, and I can come in a breakout room and we can debug it together without any problem, okay?